Hello everyone, welcome back to our session on gene interactions. And in this session, we are going to discuss the second part of uh, gene interaction, and that is non-allelic interactions. So, in allelic interaction, in the previous session, we saw that the alleles, that means the different forms of the genes, they are interacting together and they produce an effect which is different from that which was described or proposed by Mendel. And we saw what are the differences in the ratios which are being produced, which is apart from or which is variable from different from Mendelian ratios. And now we are going to see another or a entirely different type of interaction which was not at all proposed by Mendel, and that is called as non-allelic interaction. So non-allelic interaction, in the sense, it is the interaction of two different genes or two different non-allelic genes to determine a trait or a character. So in Mendelian dihybrid cross, we discussed about the interaction between two non-allelic genes, but those non-allelic genes, they were causing two different characters. For example, when we discussed the dihybrid cross, one of the gene was for seed shape, the other was for seed color, etc. So we discussed about two non-allelic interaction in dihybrid cross in Mendelian laws, but those two non-allelic genes was producing or they were determining two different characters. But here we are going to see such two non-allelic genes, they will be interacting together to produce a particular trait. That means it was earlier believed that a gene or a factor is responsible for the production of a trait. And here we are going to see two such factors or two such different genes, entirely different genes, which will be located in different chromosomes, or they may be located in the same chromosome, etc. And such genes, they are going to interact to produce a single or a particular trait. For example, flower color. To produce a flower color, what Mendel suggested was there will be a particular gene for flower color. It will have two alleles, and that will determine the color. But here we are going to see two different genes with two alleles each. Each gene will have two alleles, and these alleles, they are going. So there will be four alleles, two of one gene and two of the other gene. They will be interacting together to produce a single phenotypic trait. For example, flower color. And in non-allelic interaction, we'll be discussing five different types of non-allelic interaction. The first one, the complementary genes, the supplementary genes, epistasis, polygenes, and pleiotropism. So, in this session, we are going to discuss the first one, which is the complementary genes. So, complementary genes are two or more genes which are occurring in the same or different chromosome. The genes can be present in the same chromosome or different chromosomes, and they interact to produce a single trait. So, complementary genes, they are dominant genes. They might be present in the same chromosome or different chromosome, but they interact to produce a single trait or a particular trait, and neither of them in individual, or if they are present individually. So there are two genes and they are dominant genes, and when they are seen together, they produce a particular trait or a character. And when they are present individually, that means in the absence of the other, when one dominant gene is present and the other is absent, and the other is present and the first one is absent, in such cases they do not produce that trait. That means only when both the dominant genes are present, they will produce, or they will show, or they will determine that particular trait, only in the presence of both the dominant genes. And such an interaction is referred to as complementary genes. So, complementary genes produce that trait only in the presence of both the genes. Both the dominant genes should be present. In the absence of one of the dominant genes, they will not produce that particular character or that particular trait. And such an interaction of these two dominant genes is referred to as complementary genes. And example of this is the flower color in sweet peas. So there are two varieties of sweet peas. Here there's a spelling mistake. It's not sweet pea, it is sweet peas. One producing red color flower and the other producing white color flowers. So when two genetically different, as you see in this particular genetic cross, when you are crossing two white varieties or two genetically different white varieties which are true breeding. So these terms should be noted. They are genetically different, true breeding white flowered plants. When you cross two genetically different, you can see this. So this is homozygous dominant for one of the gene, and this is homozygous recessive for the other gene. This is homozygous recessive for the first one and the dominant for the other one. So when such genetically different but phenotypically same and true breeding in the sense if you are crossing or if you are self-fertilizing this particular plant, it will always produce white flower. The same with this particular plant. That's why you call it as true breeding. So in any generation, if you self fertilize this particular plant with white flower, it will produce only white flower, and this also does the same. That's why they are called as true breeding plants. And when such genetically different true breeding white flower plants were crossed, you get a heterozygote, and that heterozygote, it produces a purple color. So color is produced in this particular heterozygote. So F1 heterozygote is giving purple color. And when this F1 heterozygote has the normal Mendelian dihybrid cross, this F1 individual, when it was self-fertilized, you can see this the checkerboard and the normal type of gametes as we wrote in a dihybrid cross, the four sets of gametes. And the result was interesting because this particular experiment was conducted by Bateson and Punnett, uh, uh, but when they expected a ratio which was a normal Mendelian ratio, which is 9 is to 3 is to 3 is to 1, but they were surprised to have a new ratio which is 9 is to 7. 9 purple and 7 white rather than 9 is to 3 is to 3 is to 1. So they were interested to know what happened or what is responsible for this particular change in the Mendelian ratio to 9 is to 7. So 9 purple and 9 white. There were no intermediate type of individuals which are being formed. So every individual formed was either white flower colored or the other one which was purple color flowered. So the reason or when the F1 hybrids were self pollinated, the F2 generation gave a new ratio which is 9 is to 7 rather than the normal Mendelian ratio. And coming to the explanation for this particular phenomenon, that means the change in the ratio to 9 is to 7, and it was explained in such a manner. So, this particular purple color in sweet peas, the purple color flower, purple color is due to a pigment which is called as anthocyanin. So, that is a pigment which gives the flowers purple color, and anthocyanin is produced from a colorless substance. So this usually the pigments are derived through a metabolic pathway. It will have a substrate, and that will be converted in different manners, resulting in many intermediates, resulting in the product. So the product here is anthocyanin, and the colorless, it is produced from a colorless substance which is called as a chromogen, and an enzyme act on the chromogen, and chromogen is colorless. That means when chromogen is present, only the chromogen is present, the flower color will be white. And when chromogen is acted upon by an enzyme, it will give rise to anthocyanin, and that will result in purple color in the flower. So dominant gene capital C is responsible for the production of the this chromogen. So if capital C is present, uh, either in homozygous or heterozygous condition, both can be the same effect. Either in homozygous or heterozygous condition, because it is dominant, it produces the colorless substance called as chromogen. And when this is recessive, it does not produce the chromogen. And the dominant gene capital P, whether it is in the homozygous or heterozygous condition, this produces the enzyme which converts chromogen to anthocyanin. So when capital P is present and chromogen, and that means only in the presence of only when capital C and P is present, you get a purple color. So imagine a situation when capital C is present and P is recessive. Okay, so this is the case. So what happens is C is dominant here, chromogen is produced, but P being recessive, enzyme is not produced, and hence chromogen will remain as chromogen itself. The same when you observe here, here chromogen is absent, but the enzyme is present. So because in the absence of the substrate, the enzyme does not have any role, As a result, the enzyme does not have anything to convert it into anthocyanin. So the effect is, even though the cytological things which are happening inside both the plants are different, the effect is same, which is white flower colors. So here chromogen is present, here enzyme is absent, here chromogen is absent, enzyme is present. But what happens is, thus the production of anthocyanin depends on both capital C gene as well as capital P. So white flowers are produced when in the absence of either or both of the dominant genes. Either if one is present or both is present, whatever is absent, the color flower color will be white. So this what happens is, when the purple color flowers, if you observe all the purple color flowers, you can see in them at least one capital C and P should be there, which is a heterozygous condition that can also produce the same effect. But when you see all the white flower colors, you can see this is with capital P. There is no sorry, capital C and no capital P. Here is also there is no capital P. Here is there is no capital C. Here there is no capital C. Here there is no P. Here there is no C. Here there is no C and no P. So in every case of the seven, you are having white flower colors, because the absence of neither. If chromogen is present and enzyme is absent, you get the white color. If enzyme is present, chromogen is absent, also the effect is the same. And when both is absent, also the same as the effect, the result will be white flower colors. And this is also, or this complementary genes is also an explanation of another phenomenon, which is called as atavism, throwback, or reversal. That is nothing but reappearance of an ancestral character. Reappearance of an ancestral character, in the sense that it is considered that this capital C and capital P, they were the wild type in the earlier past, and they got mutated during the evolutionary process. The capital C got mutated into small c, and capital P got mutated into small c. And this mutation, or this particular traits, that is the color traits of the earlier ancestors, they appear when these two come together. That means when these two individuals they come together, they do not have neither purple color in this and purple color in this, but when they come together, an ancestral trait is being expressed, and that is why this is also taken as an example or an explanation for atavism, reversal, or throwback. That is the reappearance of a remote ancestral character. So this capital C and capital P is considered to be a remote genes which got mutated as small c and small p, and they appear when or appeared when the complementary genes interact together. So that is why it is taken as an example for atavism or reversal or throwback. So with this, we come to the end of our discussion with complementary genes. In the next session, we'll see the second type of non-allelic gene, which is the supplementary gene. Thanks for hearing.